Hi, everybody. Welcome back. This video, we're going to start digging into really the material of the class. And our initial focus is going to be on looking at problems associated with the modeling of biological macromolecules, the molecules that underlie much of protein and much of biological function, proteins, DNA, RNA, and so on. And in particular, modeling perspectives, modeling frameworks that look at them in largely atomistic or close to atomistic detail. Let me switch over to the board to be able to write on for me to get started here. Okay. So I want to start off with our discussion here, framing the biology a little bit, explaining the problems we're trying to work with, and why we think they're interesting. I'm assuming here almost no background in biology. I know that some people do have more of a background. I know some people have less. And I want to really sort of not leave behind the people who might have less of a strong background or very little background in biology. And so I'm going to start from the basics in some sense. A lot of the motivation for what we're going to discuss here and in future seconds of the class can be framed within the context of the central dogma of molecular biology. So our motivation, the central dogma. Now, what is the central dogma of molecular biology? It can be framed quite simply. This is a cartoon that you'll see in almost any molecular biology book. DNA goes to primarily messenger RNA, but other RNAs as well, which leads to protein. Right? And it encapsulates the primary modes of information flow in essentially all biological systems, all living systems. Should actually have one additional arrow here that I'm going to start now go through and labeling and kind of think about what this means. What does the central dogma really try to tell us? Why is it so central? How does it inspire problems to solve? DNA we can think of as the primary information storage molecule in a cell. DNA, the genome of an organism, contains, in some sense, all of the information required to propagate that organism. Turns out that there's a ton of emergent properties beyond what's just directly in the sequence. There's many other layers off of there, but in some non-trivial sense, the DNA does store a vast quantity of the information of what it means to be a living thing and what it means to be a particular living thing. DNA can be replicated. One of the important features of DNA, its structure, is a double helix that has two complementary molecules that can be used to reproduce essentially identical copies. And in that process of replication, it is that process of replication that underlies all reproduction of living things. The first step in reproducing a cell, in duplicating a cell, is to replicate the genome of that cell. But DNA by itself is a fairly chemically boring molecule. It doesn't have a lot of ability to function beyond sort of information storage, if you will. DNA can be transcribed. There's a process of transcription where individual genes, individual small sections of the DNA, small portions of the DNA are read out. And if we think of the DNA as being a large, many thousands of units long section, what happens in the process of transcription is that a very small, tiny section of this, known as a gene, gets not really replicated, but gets read out, and a similar molecule, but single-stranded, gets made containing exactly the information of that gene, right? And we can think of the mRNA as a transient copy of some of the information that is in the DNA. mRNA, RNAs in general, have some ability to function. There's a lot of richness in this central dogma that I'll discuss in a moment that sort of relates to this. But RNAs, well, they have some ability to sort of catalyze reactions, to engage in higher functions. They're not the stablest molecule. Their range of their functionalities is somewhat limited. And most of biological function actually ends up being carried out by proteins. And there's a translation process that reads out the information that is in a messenger RNA in this transient copy, and writes this is into a protein molecule, creates a protein molecule based on this information. Proteins, I'll discuss their detail a little bit more. Proteins generally have a structure to them. So might, for example, have some sort of structure like this, a small sheet with a helix packed on top of it or something else. But the important aspect is that we get a protein out of this. And it is proteins that carry out the vast array of functions in a cell. This picture of the central dogma is very approximate. There's a lot of missing details in here. One of the most important missing details in this picture is the question of control. These functions, one of the functions of proteins can include controlling the process of transcription, controlling the process of replication, and that feedback and the networks of sort of interactions involved in that sort of feedback from protein back into the regulation of protein expression is one of the big hallmarks of life, one of the major emergent properties that we absolutely need to keep track of. When we get into second section of the course on biochemical networks and gene regulatory networks, this is part of the puzzle that we're going to start tackling. There's another really cool thing sitting inside the central dogma. And this kind of you might get at just by asking the question of why is it structured like this? What's the point of having this transient copy? Why not just directly synthesize protein from DNA, from a DNA template? Now, there may be some functional benefits of having a transient copy. It gives you differential control and control at different time scales, and maybe that's a selectable and evolved process. But there's a lot of other interesting things around here. Whereas transcription transcription is primarily driven by a molecule known as RNA polymerase, or RNA pol, which is a protein. Again, function from proteins into RNA. Replication is driven by a molecule DNA polymerase. Again, it's a protein capturing this feedback process. But translation is catalyzed by molecules known as ribosomes. And ribosomes are primarily made of RNA. They are not protein. In addition to being catalyzed by RNA, by ribosomes, the other part, and using an RNA template, the other key piece of the puzzle for translation are molecules known as tRNAs, another RNA molecule. And so this step here, this second step of translation, is all RNA driven. It has an RNA enzyme, an RNA template, and an RNA translating machinery. And this central piece of the central dogma that is all RNA based is one of the major pieces of evidence for what is known as the RNA world hypothesis, idea that perhaps pre-life or pre-modern cellular life, in the very early stages of evolution of life on Earth, perhaps there was a world of living things entirely built around RNAs. And at some point, those RNA organisms, if you will, evolved proteins as an ability, or an ability to make proteins as a way to improve function, but still maintained sort of a core RNA replicating machinery. And then independently, who knows at the order of these, although it's given that These processes need protein, probably would have happened after the evolution of using proteins. The use of DNA as a stabler, longer term information storage unit evolved as well. And that these more complicated systems were better adapted enough, significantly better adapted in such a way that there's no remnants of those early stages of the RNA world left. This is all hypothetical. These are processes that might have taken place 3 billion years ago, something on that time frame, order of time. But it's a very plausible one. I personally think it's one of the most likely mechanisms to evolution of cellular life. Anyway, I don't want to spend too much time digging into this, so we'll largely leave it there. I will also sort of quickly point out there's a lot of stuff that can happen in biology beyond the central dogma. There is such thing as reverse transcription, where RNA is converted back into DNA. There is regulation by various other small RNAs at the translational point. There's a lot of complexity here. But for a kind of bird's eye view of what might be going on, this isn't a bad starting point. Okay. So if we think about a big part of studying biology, why do we study biology? What do we want to know? Often one of the big things we're trying to figure out is, why do things work? How do they work? What underlies function? Right? And we have this word function in our central dogma. So maybe this is a place to start, right? I've mentioned that part of function comes from how proteins are able to regulate transcription, how proteins are able to regulate replication. But what are all of the functions that proteins can do? How do we tell what a protein's function is? How is that function encoded in the information that came from the DNA? Right? How do we answer these questions? In order to get there, let's do another little mini biology lesson, thinking a little bit about what proteins are and how they work. Okay. So I've said already that proteins are the molecules that carry out most of the functions of a cell. Right? There are certainly things going on in cells that are done by molecules other than proteins. Ribosomes are certainly a major one of those, but there are many other examples also. However, by far the class of molecule that drives the largest amount of biology is, or at least low-level function in biology, are proteins. Proteins are polymers of amino acids. And amino acids are small molecules that have maybe on the order of 10, 20 atoms in each one of them. And proteins tend to be built up of anywhere from maybe 50 to about 500 amino acids long. as a general rule. There are some proteins that are a little bit smaller. There are proteins that are bigger, but this sort of captures a fairly rough idea of the size of them. Right? And this leads roughly to proteins being somewhere on the order of about 10 to the 3 to 10 to the 6 or more atoms in each protein. So from 1,000 to 1,000,000 or more atoms. These are big systems. These are big, big molecules. We're not going to worry too much at this point about, actually, maybe I will sort of give a quick overview of what an amino acid looks like. An amino acid has a basic structure of having a nitrogen, a carbon that is referred to as the alpha carbon, and another carbon attached to that. This carbon has a oxygen with a double bond towards it, and there is a group that we generally refer to simply as R. This represents that there's diversity in the amino acids. There are in the most commonly occurring situation, 20 amino acid types, which differ in this R group. The rest of the structure is the same. Almost all of them have actually, all of them do have an additional hydrogen off of there. And a amino acid by itself actually has a structure that has, at least in solution, has what's known as a zwitterionic characteristic, that it has a positively charged end and a negatively charged end. And what's really interesting about amino acids is that they can form these polymers, and what these polymers look like essentially come from taking a nitrogen of one and placing it, removing this oxygen here, so that there's now another nitrogen here, another alpha carbon, another CO group. One of the hydrogens on this nitrogen stays there, and we lose a water molecule. We take 2 of the hydrogens off, we take 1 of the oxygens off, and that makes water. And then there's an R group off of this alpha carbon and a hydrogen. And we can think about doing this in both directions or continuously. And so we then have another NH, another C alpha with another R group, and so on. So when we say that they're polymers of amino acids, we mean that we have this common structure, this NC-CO, 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 that we call the backbone. So this section here is known as the protein backbone. And hanging off of the protein are these R groups, or off of the backbone are these R groups, which we refer to as side chains. So we have a common core backbone, and then this set of side chains where each one of these can be 20 different types. Right? And it's this large number of different amino acid types that lead to the diversity in different structures. If you think about, even just for small proteins, if we think about a 50 amino acid protein, how many different 50 amino acid proteins might there be? Well, if there's 20 different choices at every position, we have 20 to the 50 different possible sequences, different polypeptides, different potential proteins. So we have this huge richness of possible structures that we can get out of this heteropolymer of amino acids. Now, strictly speaking, any polymer of amino acids like this, every polymer of amino acids is not strictly a protein. The term we use for an arbitrary sequence of amino acids linked together in this way is a polypeptide. Poly for many, peptide because the bond formed between these two units, the CO and the NH, when we removed water here, that's known as a peptide bond. So many peptide bonds. What makes a protein distinct from a polypeptide? Well, we have these many, many different possible polypeptides. Not all of them will carry out a function. What characterizes a protein? A protein has a function. A protein is a polypeptide that has been evolved or in recent decades engineered or designed, but a polypeptide that has a well-defined function associated with it. Most proteins, in order to function, also have a particular structure associated with them. And this leads us to our next important aspect of protein biology, the question of protein folding. Functional proteins, functional polypeptides, usually fold into a well-defined three-dimensional structure. So I'm not going to keep couching the words most, many. I'm going to be a little bit sloppy and say strictly that proteins fold into a well-defined three-dimensional structure. There are classes of proteins that are referred to as intrinsically disordered proteins that don't have structure in the same way that I'm going to talk about them here. But let's think of those as a special class that doesn't really affect our discussion here. Proteins fold into unique (3D) structures. What do we mean by that? Well, if we think about just a polypeptide, the cartoon that I drew of it on the previous slide here looked kind of linear, but there's flexibility in many of these bonds. And if these various bonds are flexible, if they can move around, what you might expect of a random polypeptide, if you make a polypeptide out of random sequences, you might expect it not to have much structure. And so you often might see a cartoon pictured here of kind of just a squiggle. And the idea here is that a random polypeptide would have kind of no real discernible structure. And actually, this might be what it looks like in one snapshot in time, but it would move around into many, many sort of arbitrary or random shapes over time. Now, there's some physics underlying that. You can characterize what that distribution of shapes might be, but it's not a structure. Right? What happens in many, many proteins is that they fold into often compact, sometimes elongated and other sort of structures, but unique, well-defined three-dimensional structures. You can look up all sorts of beautiful pictures of these, but this is done through a process of forming, first of all, secondary structure. And I don't want to imply a mechanism here, but portions of the structure may form particular arrangements of the backbone. So we might have what's called a beta sheet as one common form. We also may have portions of the structure that form what are known as alpha helices. And I apologize, I am far from an artist. We also have alpha helices. These are what are known as secondary structure elements. And actually, formation of secondary structure is probably linked to the folding that I'm going to sort of mention now, but we can conceptually break it down into the sort of ideas that we have just the amino acid sequence, which is primary structure, these sort of local types of structure that are secondary structure, and these secondary structure elements kind of collapse in on one another. And so one possible structure would be having these two beta sheets pack against one another, and then have these pair of helices, maybe one helix sits on top of those, and the other sits underneath those to give sort of a sandwich of alpha helix on either side of a beta sheet in between. Right? And this kind of unique folded structure is characteristic of proteins. Once a protein has folded into a structure like that, it tends to not change very much. It stays in that structure for extended periods of times. Some proteins do have important dynamics where there's some changes, but they tend to be much, much more restricted than the kind of random coil structure that we have here. The three-dimensional structure or tertiary structure, this is often referred to as the native state of the protein, is essential for function. The random coil structure is not functional. A polypeptide that had all of the secondary structure elements properly formed, but without the proper tertiary fold, would still be non-functional. Only the native state has the proper function. Now, this picture already leads us to two big grand challenge problems in biology. And so let me highlight those here. And these are going to be motivation points for our first set of modeling exercises that we get into. What are the two grand challenges that we can mention first of all? The first is the question of how sequence leads to structure. This is often referred to as the protein folding problem, or the protein structure prediction problem. It turns out that doing experiments to figure out the structure of proteins is hard. Doing experiments to figure out the sequence of proteins is easy. The reason that it's easy to find out the sequence is because that sequence comes from DNA, and there's a very, very powerful experimental tools that are fast and cheap that let you determine the sequence of nucleic acids. And so it's easy to find protein sequence generally. Protein structures need to be solved with much more involved methods. Some of the techniques that are most commonly employed are X-ray crystallography, where you first of all have to somehow crystallize your protein in a high quality enough form. That's a very, very, very challenging thing to do. Some proteins crystallize easily, many proteins don't, a major limitation of that method. Other methods include things like NMR spectroscopy, cryo-electron micrography, and so on. But the experiments are hard, they're expensive, and they don't always work. And for that reason, our knowledge of protein structure greatly, greatly lags our knowledge of protein sequence. And if we could eliminate that gap, if we were able to consistently predict the structure of proteins, it would be a major, major benefit. We can also, actually before going on, we have two names here, protein folding, protein structure prediction. These are both kind of, there's two questions we can actually ask here. There is a how question and more of a what question. There is a mechanistic question we might ask. That is, how does sequence dictate structure? What is the physics that drives that process? Can we understand that physics? Can we simulate that physics? This is really what the protein folding community has aimed to try to do. Can we understand the process of protein folding? There is another class of problem within this problem here that is simply the predictive or what problem. This is more often referred to as the protein structure prediction problem. These are not exactly the same thing. I mentioned in the first lecture how we have to think about different ways of looking at the same problem, and sometimes one way gives us different insights than another. It is certainly true that if we had a complete, perfect mechanistic understanding of protein folding, that we knew exactly why all proteins fold the way that they do, and models that could simulate that process, we would have an infrastructure that could allow us to do prediction. It might not be fast, it might, but it would work if our understanding was complete. We do not need, however, to necessarily have a complete understanding of how proteins fold in order to predict protein structures effectively. And there are algorithms for predicting protein structure that rely very little on any mechanistic understanding. And there are approaches for protein structure prediction that, and I should have said prediction here, I wrote down the wrong word here, protein structure prediction, sorry. There are methods for solving the protein structure prediction problem that do rely on mechanistic details. We'll dig into these more once we get into the more algorithmic side of things. So one grand challenge is this idea of how does sequence lead to structure? The other big grand challenge in this first piece is how does structure lead to function? Even if we had a complete perfect solution to the structure prediction problem, that does not mean that we would be able to kind of read directly into function from sequence. Structure is a prerequisite for effective function, but how is that encoded? What is it about the structure of a protein that leads to its function? There are a lot of different sort of sub-problems within this. Some of the things that impact function are molecular associations. Molecules, many biological molecules have an ability to physically interact with one another, really to kind of stick to each other, sometimes for only a short period of time, sometimes for longer periods of time, sometimes undergoing a chemical reaction after they have stuck together, sometimes not. But as a starting point to understanding function, if you can understand what molecules is this protein able to physically interact with, what molecules can it associate with, you're part of the way toward understanding function. Another important aspect of protein function is understanding catalysis. Many proteins lead to specific chemical reactions or catalyze specific chemical reactions, often after a molecular association step. Um, and proteins can have differences. The same protein might be able to bind, physically associate with two different molecules, but can only effectively catalyze a reaction for one of them as opposed to another. So understanding and being able to predict the ability of proteins to catalyze reactions is another part of this. Another important part of the functional piece is the concept of conformational dynamics. So I just said proteins often typically have a well-defined structure. We say that it has a structure because it doesn't change very much compared to the diversity of structures in, say, a random coil of an unstructured or a disordered protein. That doesn't mean there's no motion. Even the most stable protein still undergoes small molecular motions. And differentially large motions in different parts of the molecule. Those motions are often essential to function. They can be important for catalysis. They can be important for allowing a single protein to associate with a larger range of different molecules, because there's a little bit of plasticity in the shape of the protein. And so understanding the conformational dynamics of a protein is another major part of this puzzle. Right? So two big problems we're faced with. How does sequence dictate structure? And how does structure dictate function? With both of these, and the various sub-problems within the structure-function problem, we can always break down the problems into both a mechanistic aspect, a question of how does something happen, more scientifically motivated question perhaps of what's going on and what's the physics or the chemistry that drives these processes, and more predictive questions of what happens, perhaps not worrying about the details as much. In the same way that we can predict the structures of some proteins without necessarily understanding how they fold, the mechanism of their folding, we may be able to predict some aspects of molecular association, some aspects of conformational dynamics, perhaps even some aspects of catalysis without understanding the mechanism. Some of it we also may be able to use mechanistic understanding to make these predictions, and we may need mechanistic understanding for some of those. But there's the two angles that we should always keep in mind in pursuing the problem. Okay, I think that in the interest of time and in your own sort of attention span, I'm going to end this particular video here with these two grand challenges that we've presented. What we're going to be doing in the first section of this class is starting to develop the models that we need to tackle these two grand challenge problems. We are going to give a little bit more biology about each one as we do so, in order to kind of put together the puzzle. But we'll also be incorporating the model building aspect, the physics aspect, the mathematics aspect, and the numerical methods aspect. Until the next video, take care and be well.